This Week in Virology, the podcast about viruses, the kind that make you sick. Hello, Cold Spring Harbor! Or maybe I should say, hello, Retrovirus. Oh, that doesn't get as loud a response. Interesting. All right. From Microbe TV, this is TWiV, This Week in Virology. A special episode, recorded on May 23rd, 2018. I'm Vincent Racaniello, and you are listening to the podcast all about viruses. Today, we are recording at Cold Spring Harbor Laboratories, the first TWiV at Cold Spring Harbor Laboratory. We're at the 44th annual meeting on retroviruses. And it's the first for the retrovirus meeting as well. And joining me today, my co-host of many years, Dickson Despommier.
Hello, Vincent.
Welcome.
And everybody else. It's a pleasure to be here. This is actually my first visit to your august institution. And I am already impressed with your dinner.
Dickson has low standards, right? I think. Well, Dickson and I drove out here just now, and we spent 4 hours in the car together. You know, I can barely get through 2 hours with you on TWiV, right? We have a couple of guests who are all attending the meeting, and on Dickson's left, for his second time on TWiV, from Tufts University, John Coffin.
Hello again, everybody.
Welcome back, John.
Thank you very much.
Glad
to be here.
John was on TWiV 320, which was called Retroviruses and Cranberries. We talked a lot about cranberries.
I didn't know you could catch it from cranberries. This is new to me.
It's amazing. It's amazing. Our next guest is from the National Cancer Institute. It's his first time on TWiV. Steve Hughes, welcome.
Thank you. It's a pleasure to be here.
And then we have two younger guests. We're all older, except for the last two. Yes, you are young. Yes. You should enjoy it, because it doesn't last. We have from Yale University, Yachi Ho, welcome to TWiV.
Thank you. Hi, everyone.
And from the Laboratory of Retrovirology at Rockefeller University, Matt Takata, welcome to TWiV. And we also have a nice audience. Are you all retrovirologists? Or is anyone not a retrovirologist?
Yeah, well, that's me too. I'm not even a virologist.
Dickson is not even a virologist. So I have to say, Dickson started TWiV with me almost 500 episodes, 8 years ago.
That's true.
And he's not a virologist. But listen to this. Dickson, how many genome types are there?
7. And you're supposed to put something in my mouth now to reward me for knowing that answer.
No, it doesn't work that way.
So, but I must inject here a credential, and that is that I got my degree at the University of Notre Dame working on germ-free animals, supposedly germ-free, but of course we know now that perhaps they are not germ-free altogether. But I was in the laboratory that Dr. Morris Pollard ran. So he was an expert on rabies and psittacosis. So I heard a lot about viruses while I was a graduate student. So perhaps some of it rubbed off.
Well, you've also heard of almost 500 episodes.
Exactly right.
So I hope you absorbed something. Although sometimes we all make mistakes, right? All right, so the composition of this panel is done for a reason. So the two younger people have won prizes at this meeting, and we'll come to that a bit later. We'll talk about their work. But we have these two senior gentlemen here who are going to tell us a little bit about this meeting, because presumably they've been to a lot of. This is the 44th meeting. So, John, have you been to every single one?
I have. I'm the only person who's been to every single one, actually. There are a few that have been to almost every single one, but missed some for some feeble excuse or another. And I was at two. before this meeting, there was a tumor virus meeting that started actually, the first of these was in 75. There was a tumor virus meeting that covered both DNA and RNA tumor viruses that started in the late 60s. And I was at 2 of those in 70 and 71. And then at all of these starting in 75 up till now.
75, it started as the retrovirus meeting?
Started as a retrovirus meeting in 75, or split off from the all tumor virus meeting. It used to be that the DNA tumor viruses would come first, and the retroviruses, then RNA tumor viruses, would come second. And in between, you could hear the sound of all the cars peeling out of the parking lot of all the DNA virologists who didn't care at all about retroviruses at the time.
So it made sense.
They
to
wondered why
separate
anybody
them.
should care about
Yeah.
retroviruses at the time. It became apparent about 1970.
Have you been to everyone? No.
No, I'm much younger. I only started
Only in
coming,
years.
I only started coming in 1977, so I'm your basic newcomer.
He's the new guy.
Okay, so 77 retroviruses. So by then, reverse transcriptase had already been discovered, correct?
1970.
1970, so that's why you could call it a retrovirus and not an RNA tumor virus meeting.
That actually more or less came with the advent of HIV research in the 80s, when it was recognized that there were really good reasons to study retroviruses that weren't tumor viruses. And the name was changed in part because the focus of the field changed from viruses that cause tumors and carried oncogenes, or activated oncogenes, and into the human pathogen HIV.
That brings up a point I wanted to explore, and this is a good time as any. Everybody here is working on HIV, all 4 of you, right? Is that correct?
I work partly on HIV and partly on
Cranberry.
cranberries, and a large part still on virus evolution and ancient viruses that inhabit our genomes.
Endogenous retrovirus.
Endogenous retrovirus.
So in the audience, raise your hand. Who works on HIV?
Almost every.
Good Lord.
All right, put your hands down. Who works on a different retrovirus?
Wow.
That's
Still
pretty
some.
good. So there's still some. Rouse sarcoma virus? Right.
Little injection here. I spent 3 years at Rockefeller. as a
Avian
postdoc.
sarcoma viruses? Yeah, there you go. Walleye dermal sarcoma virus? No fish retrovirus. What am I missing?
Koala, yeah.
Koala. Okay. You work on all those? Okay. All right.
Yeah, well, there's always one.
Why are you here? You know, I have to tell a story. My first Cold Spring Harbor meeting, I was a postdoc with David Baltimore, so it might have been 1980. And there was a meeting on mutagenesis of DNA, because the techniques were just beginning at the time. And I worked on poliovirus, and people would say to me, "What are you doing here?" Because everyone thought you couldn't mutagenize RNA, and they're right, of course, but we had made a DNA copy of it, and so that's why we were here. So maybe there's a good reason for you to be here. Now, so obviously people work on other retroviruses, as you can see, and I was wondering about that, because if it had been all HIV, I was going to ask, is that a good thing that everyone works on HIV? But it's not the case, right, John?
Yeah, well, HIV has become a paradigm, really. I think probably in some respect for almost all viruses, and a substantial chunk of immunology as well, just because of the suddenness and importance of the problem. So that much very fundamental virology is done on HIV now, that not necessarily directly focused toward understanding HIV disease or treatment.
Now let's have a hypothetical, Steve. Going forward, we get rid of HIV. So we're about to get rid of polio, which is the virus I worked on my entire career, and we can't work on it anymore. If that happened for HIV, would be great if it did, right? Would other retroviruses expand to fill in the space?
I don't think so, but before we go there, I think I would say that although I would very fondly hope that we can get rid of HIV, I think the current numbers are very much against that, in part because we have more new infections each year than we have deaths. The actual number of people that are infected is increasing both in the United States and worldwide. And I think the prospects for eliminating the virus are not good. If nothing else, because we have a substantial number of people worldwide who are infected and don't even know they're infected. And I'm sorry to tell you that, but I think that's the answer.
But it was a hypothetical.
I think the only other significant human pathogen that we know about is HTLV, which is much less pathogenic than HIV in terms of the fate of the people that are infected. But there have been quite recently a recognition that there are places in the world where much higher fraction of the population is infected than we had previously anticipated. I wouldn't, if nothing else, because the pathology is not as dire, expect that it would take the place of HIV in terms of research, but I certainly think it deserves research attention. Large numbers of people are infected and people do die of the disease.
So let's take it from another viewpoint. When we first discovered HIV, one of the reasons we could develop antivirals so rapidly because we had this huge knowledge base from studying other retroviruses. So I guess the question is, should we really be doing more with other retroviruses and being prepared for something else?
I think to a considerable extent what we do with HIV feeds well into that, although there obviously are big differences between HTLV, HTLV1, and many of the other viral pathogens, particularly of animals that we know about. But a lot of the knowledge base that we get, as it was in the case with HIV, I think is applicable to getting, to getting at other retroviruses.
So what about the younger people? What do you think, Yachi?
I think that we should work on HIV as a model, which will help us to solve lots of other problems. For example, I got this prize called Andy Kaplan Prize, who was a physician who worked on the pathogenesis of HIV. So he's a physician, so he sees people are suffering. At that time, little is known about HIV, so he devoted his career into this. I came in, there's art, and people get treated very well. However, there's no cure, and people are still fighting against stigma. So then it's a perfect time for us and many young scientists to come to this, to get to know many, many details about HIV. With all these technologies, this will actually help us to guide the understanding and therapeutics of other viruses, not just retroviruses, but also other viruses. So I'm very optimistic.
How many times have you been to this meeting, Yachi?
This is my first time.
First time. Mm-hmm. Tell us your training. Where did you go to college?
So I got my MD in Taiwan. I got 3 years of internal medicine residency and 2 years of ID fellowship. I practiced as ID physician for 1 year. When I have clinics in the prison that I see HIV-infected individuals, and I actually really love them because they got HIV for a reason. They may not have any choice. So we can help them, and I'm very grateful about this experience. However, I thought that if I continue to practice for 30 years and I may help all the patients that I see, but I still don't solve this problem. And I hope that there's other ways that you just change one little thing that could help many, many, like millions of people around the world. And also, I'm very interested in basic science. I've been working on, say, Staphylococcus aureus and influenza and other kind of stuff. So I feel like I should use this basic science tools to solve these clinically important questions.
So did you do a postdoc or
I did my PhD at Johns Hopkins with Dr. Bob Silicanos. I got my PhD there and got a short postdoc for 1 year and went on to do a little bit more semi-independent research until I recently relocated to Yale University in September last year.
Brand new professor, huh?
Newbies.
Congratulations. So despite working in an HIV lab, you didn't come to this meeting until this year, right?
I was so afraid that if I don't understand all these little details, I'll come and ask very stupid questions. So I prepared this for 9 years until this year.
There are never any stupid questions. Matt, is this your first retrovirus meeting?
No, this is my third.
Wow.
My third time here, yeah.
And you're a PhD student, right?
I am.
Are you almost done?
Yeah, I'm a fifth-year student in Paul Binash's lab.
Okay. And what are you going to do next, do you know?
I'm trying to figure that out now. I'm still doing some follow-up research to my current project, but I'm also looking at different labs to do a postdoc in.
So John, can you remember an amazing discovery that was reported at this meeting?
Oh, I think there have been a number. The discovery of HIV was reported here, but not at this meeting. A lot about, well, one of the most intractable problems in retroviruses for many years was the mechanism of integration, DNA integration, because it's really quite different from almost everything that we know about. And it was known on very good evidence that it was happening and about the properties of it, but there was no system, really good system for studying it. And a lot of the work, the very first work on developing the tools to actually look at that was presented here first, for example. And a whole lot of the really fundamental stuff in retrovirology. The discovery of reverse transcriptase was almost reported here, but it was actually at the symposium, it would have been a week or two after this meeting.
Okay. So integration, you think, is a big
integration is a biggie. There's a lot, actually. Some of the first identification of retrovirus receptors was reported here. Actually, one of the things that was reported here first in public for the very first time at this meeting was the discovery of splicing in eukaryotic messenger RNAs. Although that was not in retroviruses, but the talk about that
really?
by Rich Roberts was actually at this meeting.
Cool.
As a few of you here might remember.
Yes.
Steve, do you remember any notable things announced here?
If you go back far enough, this meeting, in a sense, derived from the RNA tumor virus meeting, and a great deal of what we know about oncogenes was, in fact, first reported here. Although a lot of it wasn't necessarily presented here first, it was, a great deal of information about structure and function of retroviral proteins was reported here early, if not first. Things like the structure of the provirus, the way DNA was synthesized, the fact that many of the structural proteins of the virus are synthesized, and the enzymes are synthesized as polyproteins and how they're processed. The original sequence of the first provirus. The history of the meeting, in a sense, is the history of the field. And this is, it's less true in the clinical sense. But in terms of the sort of the basic biology and the basic molecular biology of retroviruses, if you went back through the collection of the retrovirus abstract books, you would have the history of the field.
I mean to add one thing. If you go back to the tumor virus meeting, the use of restriction enzymes for DNA mapping was first reported here.
So why Cold Spring Harbor and not somewhere else? Do any of you know?
Well, Cold Spring Harbor had a very long history of cutting-edge meetings in all kinds of biology, molecular biology, and genetics, going way back. I mean, the symposia go back, I don't know, 100 years or something, and so they'd had a tradition. I mean, running meetings like this and courses associated with them had been a part of their mission, really, since the very first days of this laboratory.
There was a time at which there was a very strong conflict at the NIH about whether you should use viruses as paradigms for human cancer, and whether or not, and at the time, although it seems odd now, everyone knew that viruses did not cause cancer in humans, which is of course wrong, but it didn't matter. That was dogma. And in particular, there was a very strong feeling on some people at the NIH that the idea of using animal models in which you induced cancer in the animal, that was irrelevant to the idea of studies that had some meaning for studies of human cancer. And one of the things that Cold Spring Harbor did was to advocate for the study of tumor viruses as a tool. And they advocated locally in the sense that much of the research that was done here was on DNA tumor viruses. But they were equally strong in supporting the meeting which devolved from this originally joint RNA and DNA tumor virus meeting for supporting a meeting on RNA tumor viruses. And the current retroviruses meeting devolved from that when the study of oncogenes split off into a different meeting about 1980.
So let's try another approach. John, do you remember anything notable that turned out to be wrong?
Well, the answer is for sure. In fact, I was the organizer of, one of the co-organizers of the 1981 meeting, at which the highlight of the meeting, or the low light of the meeting, was a talk by a graduate student. Should I give his name?
Mark Spector.
Mark Spector. How many people in the room know who I'm talking about? Okay. For those that are listening, it was maybe 15% or so. It's not a huge fraction. So Mark Spector was a graduate student. He worked up at Cornell, and he had discovered, if you like, how kinases were connected to one another in both signaling, in ATP transport, and in oncogenesis. And he created this beautiful house of cards. And if anybody send me an email if you want to see, I'll send you a copy of the cover from this meeting, and in which he shows all of these the PK whatever is all sort of linked together to one another. And he would go around. He was spending a lot of time going around from one famous oncogene lab to another to another to another. And I won't name the individuals, but whose lab he went to with his tools, with his toolkit to show how their particular oncogene fit into this beautiful hierarchical scheme that he had developed. And it turned out, and in fact, it turned out, to make a long story short, that what he knew what size everybody's protein was supposed to be, and he knew that they were all supposed to be kinases. So in principle, if you labeled them with P32, you could see what they were, you could figure out what their targets were, and so on and so forth. And so he would go to everybody's lab, and he would take his magic antibodies, and he would take his magic stuff, and he would run gels with them, and sure enough, he'd come up with these P32 labeled bands of the same size, of the size that corresponded to that person's oncogene, and he would fit it sort of beautifully into this beautiful scheme that he had made. I mean, it was all, a lot of people were really, really taken. In fact, there were well-known labs were fighting over this guy to have him come and work on their protein. And he was getting job offers like crazy as a graduate student before he'd even begun to write a PhD thesis. And it turned out that one of his thesis committee members, a very well-known retrovirologist actually, who's not here unfortunately right now, got a little suspicious. Well, he gave a talk at Cold Spring Harbor. He actually became the subject of a retro trivia question, is who lists the people who have been given more than 10 minutes to talk at Cold Spring Harbor. He was actually given a half hour. It was at the end of the Friday session, so anybody who wanted to leave could, but very few did actually. And it was when these meetings were across the street in the Bush Auditorium where the posters are now, and it was very hot that day, as I recall. The air conditioning was terrible. And this faculty member got really fairly suspicious about it, and he went back and did a scintillation spectrum on the bands that this guy was getting in gels. Turned out what he had called P32 was actually I-125. They went back through the purchase records and found out that what this guy was doing was when he was going to a lab, he knew what the size of the protein that he should be getting. He ordered from Sigma a protein of that size, labeled it with I-125, and there was this phosphoprotein, used that to salt into the gels. It was an incredibly elaborate, it was the most elaborate fraud you can actually imagine. And so I think that's probably the most famous thing that was wrong here, and it was more than just wrong. I mean, it was really it was absolute blatant fraud once it was uncovered.
But what he made up actually turned out to be correct. Right?
Well,
yes.
in some ways, none of the details were right. Sort of the overall concept that these things sort of communicated with one another and worked by kinasing, labeling, to some extent worked by phosphorylating one another, is in fact a good part of the oncogene story. It's not the entire story, but it's a good part of the oncogene story for sure.
So Steve, do you remember anything besides Spectre?
Well, I remember vividly watching him present, and there were reasons to be skeptical, but he claimed to be showing that the proteins in question were labeled with, the phosphate was on tyrosine, because of course many of the oncogenic proteins are not only protein kinases, but phosphorylate tyrosine. And he showed what he claimed to be thin layer chromatographies of the position at which the phosphotyrosine was supposed to run on a thin layer chromatogram. And unfortunately, in terms of the data, the spots on the thin layer looked like black dimes. They were perfectly round, and anybody who's run thin layer will tell you that there's always a little bit of smearing, and the top of the chromatogram never looks perfectly round. And I was somewhat skeptical before I saw for a variety of complicated reasons. But when I saw the chromatograms, I was certain that those had not been, the spots had not been run up the gel. In fact, if he'd just run them on the gel a little bit, it would have been much harder to have known that.
I'm surprised neither of you brought up XMRV.
Well, that's number 2, I would say.
Was that announced here at some point, or?
It was presented here, but it was not the first presentation. That was elsewhere. What was presented here was from a collaboration actually between my lab and Vinay Pathak and some others, was sort of the solution to the issue, describing exactly where the virus came from.
Right. All right, let's get more positive, right? Matt, you're the recipient of the UTA Prize.
Yes.
I presume you know what that is, right?
Yes.
What is it? Tell us.
It's in honor of a former participant of the conference who unfortunately passed away, but contributed much to the field of retrovirology.
And why did you get it?
I got it because of the thesis work of my PhD.
Okay, so it's for a student, right?
Yeah. It's for the graduates.
So you're at Rockefeller, you're a PhD student. Where were you before that?
I was in Minnesota. I was in a small private school in Minnesota studying marine biology. Mm-hmm. Yeah, it's a little far, not even marine viruses. I was just studying
in Minnesota.
Sea urchins. Yeah, I was a marine biologist, the furthest place away you could possibly be from the sea. A little ironic, but
it's okay.
got this.
Bob was from Minnesota too, right?
Yeah?
Sure.
Okay, I believe. I'll find out. You're going to get an email saying that.
No, he's from Minnesota.
Okay.
Robert Zimmerman.
Robert Zimmerman, right. Exactly. So what brought you to New York City then?
Rockefeller University.
Yeah.
It seemed like a good time to be living in New York City. Rockefeller was a fantastic school to be at. And so it just worked together and ended up there. I knew I wanted to transition to something a little bit more relevant to human health than sea urchins and their immune systems. Rockefeller seemed like an all right place to be doing that. I didn't really know what I wanted to work on, but.
So can you name a famous retrovirologist who got a PhD at Rockefeller?
Who got a PhD?
Shameful questions.
Who knows?
He worked.
Yeah, David Baltimore.
Baltimore.
Yeah.
Yeah, Baltimore got
I
a
questioned myself.
Baltimore. Rouse was a faculty member at Rockefeller.
Yeah, no, because the PhD program didn't start until the 1950s.
Yeah, but David Baltimore is who I'm thinking of. Yeah. What department was he an undergrad in? Oh, you're talking about someone else. Okay, so Rockefeller brought you there, and then how did you decide? What, his PhD in 3 years? Yep. All right, we have a peanut gallery here. But it's okay, I like it. We should get you microphones. All right, so you were attracted to Rockefeller, whose reputation extends to Minnesota, apparently. Right?
Yeah, across the country, yeah.
How did you end up you're in the BNash laboratory, right?
Yeah.
How did you end up there?
Well, so I actually originally came to Rockefeller thinking I was going to study cancer. It's kind of the human health thing to study right now. But in the end, I really didn't find myself liking particularly any of the cancer labs that were at Rockefeller. But towards the end of one of my rotations, I saw Paul give a talk. It was actually on ApoBic that really drew me in. I've never heard of it before. It blew me away. And just hearing him talk about ApoBic, hearing him talk about tetherin, this idea of restriction factors, really, I found amazing at the time. And so I did a rotation in Paul's lab, never even really thinking about viruses previously. And here I am now.
Okay. Now, Theodora gave us, so we're revising the virology textbook right now, Principles of Virology. So she gave us your paper, which was published in Nature. I think you're aware of it, right?
Yes, as I recall, yeah.
CG dinucleotide suppression enables antiviral defense targeting non-self RNA. So it's a possibility that might end up in a textbook, which is pretty cool.
That would be amazing.
What do you think about that?
That would be amazing, yeah.
Yeah, so what is GC suppression? What is that? Explain us. Especially for Dickson, right?
Yep, talk to me.
Yeah, so it's vertebrate genomes and the viruses that infect vertebrate genomes have been long known to be CG suppressed. And in the case of vertebrate genomes, we think we know why that's the case. It's because of CPGs are methylated and then spontaneously deaminated. And over the course of evolution, you get this depletion of CPGs and the accumulation of TPGs, which we see. But when it comes to viruses, it's been unexplained why they would be subject. They're not subject to the same pressure. So it's never been understood why viruses have a suppression of CPGs until this paper that we published.
But why does this happen? Do we understand? I mean, you say it's the methylation and so forth, but
why does it happen in vertebrate genomes?
Well, first of all, it's only vertebrates, right?
Yeah. Yes.
Do we know why it's not in non-vertebrates, for example?
No, I don't think we know why it's not.
But in vertebrates, why does this happen? Do we have an understanding of why it's necessary? It's just the methylation that ends up.
That's the current understanding. Yes, we don't, and that again is just a hypothesis. That's,
All right. So
that seems to fit the data.
Both DNA and RNA viruses show this suppression as well.
Yes. Yes.
Okay, and HIV clearly does.
And yeah, and HIV is incredibly suppressed for CGs.
So tell us what you did in this paper to look at that.
Yeah, so we originally started making, we just completely mutagenized the HIV genome as much as we possibly could. And essentially we were just trying to see if we could disrupt any RNA elements that hadn't already been discovered. So there's a chance nothing was going to come out of it, maybe, because we left everything that's already been published unmutated. So we made pretty much the maximum number of mutations we could. We made 16 different viruses to study, and then we began studying them. So about half of them were totally fine. Half of the defective mutants had splicing defects, but then we had this one group of mutants that we couldn't explain why. They didn't have any splicing defects, but for an unknown reason, they were completely replication incompetent. They just didn't spread. And so, but then also, if you were to try to map the defect, you can never map it. You divide up the virus, and the defect slips through your fingers. So around this time also, though, there was this idea that you could add CGs to RNA viruses and greatly attenuate their replication. And so the thought occurred to us after banging our head against the wall for a while, trying to figure out what's going on with these viruses. Well, maybe we might have increased the CG content of them. And it turned out that we had. We had actually dramatically increased it. Yeah.
So you didn't start out to study CG suppression?
No, absolutely not. It was completely coincidental too. We made a lot of mutations, and because HIV is so CpG poor, it's actually very easy through a synonymous mutagenesis to add a number of CGs to these viruses.
Okay, so you restore CG to the genome and the viruses are crippled, essentially.
Exactly.
All right, so then you did something next, which is really interesting. Tell us about that.
Yeah, so the attenuation of these viruses through adding CGs is I think already very interesting, but it has already been published on. But the story really starts getting good when we bring in ZAP. And so ZAP has been a known antiviral protein. It was discovered in the Goff lab in 2002. It restricts MLV. But for the past 15 years or so, they've been trying to map the RNA region or some sequence that ZAP binds to, but no one's been able to do it. All they could tell was that it's these long regions of RNA. And as they would begin to divide those regions up, they would see that the ZAP effect would go away. So looking at these papers, the thought occurred to us at some point, well, maybe ZAP, this seems to be a very similar phenotype. They can't map this defect. We can't map our defect. It seems to be cumulative. So we knocked down ZAP, and all of a sudden, our CG high viruses were replicating just like the wild-type virus again. With the knockdown of ZAP, we had completely rescued these otherwise previously dead viruses.
So does this apply to other viruses as well?
We think so. So if you look at viruses that are or aren't sensitive to ZAP, you actually have a very clear divide between the sensitive viruses that have been previously looked at through other labs. The Goff lab, the McDonald lab, and the Gao lab. They've all been studying these viruses, but haven't noticed that they are actually incredibly rich in CGs, where the non-sensitive viruses are relatively suppressed for these CGs. So we haven't added CGs to any other virus and attenuated them, but it seems to be a phenomenon that is applicable to other RNA viruses.
So ZAP is a cell protein.
Yes.
So why do cells have CG suppression when they have ZAP that could take care of it, right? So ZAP corrects the problem with CGs in the HIV genome. So why doesn't it correct the cell problem?
Yeah, exactly. So it seems that ZAP has taken advantage of this low CG background in vertebrate genomes, specifically in mammalian genomes is where we know ZAP is active. So by having a really low background of CGs in the cell, it's able to then recognize what is a non-self RNA. So if a virus comes in from a like an alpha virus, for example, it's able to recognize through that CG content that virus is non-self, so to say, and degrade it.
So ZAP binds the CG?
Yes, yeah. Through CLIP-seq analysis, we've showed, I think, very clearly that the only motif, we've tried to do more complicated analysis, but really the only motif that we can find that ZAP binds is a CG. There's nothing before it, there's nothing after it. It really just seems to be it recognizes those CGs.
And what's the consequence of that binding?
So I could generally say destabilization, but that's kind of an intentionally vague word right now because we don't really know. We don't know if it's actually chopped up by a ribonuclease. We don't know if it's just sequestered or retained in the nucleus.
Are you continuing to work on this?
Yes, yeah. This is what I'm working on now. This is what I'm trying to figure out.
Okay.
What exactly happens if there's a ribonuclease, if there's something else.
So you said before you're not sure what you're going to do next, right?
Yeah.
Do you think it'll be virology or maybe something entirely different?
I'm not. I'm thinking really broadly right now. I'm not sure. I definitely want to stay within the field of microbiology, but even that's a huge field, obviously. Whether I stay with viruses or not, I'm not sure. But I'm considering a lot of options right now. Yeah.
So Dickson, it's a neat story, right?
Fabulous.
So we would have done this on TWiV, probably. But now we did it here, right?
We are doing it on TWiV.
Oh, that's right. You're right. Okay. Congratulations. Good luck.
Yeah, thank you.
Yachi, you're the Kaplan Prize winner. Who was Kaplan?
So his name is Andy Kaplan. He is a physician who got his undergrad at Columbia and went on for medical training, and then started his research training as an ID fellow in Ron Swanson's lab at UNC, and later on started his independent career in UCLA and then moved on to UNC.
And why did you get this Kaplan Prize? What is it for?
It's for a project I'm working on called aberrant transcription of HIV-driven aberrant transcription, which contribute to HIV persistence. So in this study, it's like this. So we have been working on how HIV persists in patient samples. It is actually pretty challenging because in HIV-infected individuals under long-term antiretroviral therapy, although they have antiretroviral load in their blood, but still they are never cured. Once they stop taking antiretroviral therapy, the virus will rebound. But this is very, very interesting. So we want to understand the basic mechanism how this happened. So we want to just take patient blood and study how HIV persists. There's a problem. The problem is, first, there is only 1 to 10 million per CD4 T cells per million that contain an infectious HIV, and around 1,000 per million CD4 T cells containing a defective HIV, and the rest, like 999,000, are HIV infected. So it's very hard to pull out all these infected HIV to study their mechanisms, and there's no accurate marker which can pull them out. So when I was at Johns Hopkins, this is my R21 as a project, we're trying to identify HIV-infected cells from these patients using this technique that we developed called HIV RNA sort-seq. Using HIV RNA-specific RNA probes, we can isolate HIV-infected cells from these patients. They are really, really rare. Think about 1 to 10 million. We typically sit through the sorter for the overnight and just get maybe 20 cells out of this whole thing because it's so rare. But after we sort them out in single cells, we can do a very deep RNA-seq to identify where HIV is. But this RNA-seq that we do is not just, oh, look at transcriptome, higher and lower, heat map them, or T-SNP plot them. Instead, we want to know how HIV interacts with the host genome at the RNA level. So we look deeper into the very sequence junction and found that they are, from these clinical samples, they're HIV integrated into cancer-related genes and somehow change the behavior and expression level of these cancer-related genes, such as like BOT2 and FAT3, which has been thought as a gene related to HIV persistence in patient samples. So we can use single-cell RNA-seq to look at these HIV and host interactions from the very rare HIV-infected cells from patients.
So how does this work? Explain. So it's an RNA probe that you're using, and how does it pull out the cells that you're looking for?
So we take resting CD4 T cells from patients. They're all under suppressive antiretroviral therapy, so there's very little HIV RNA transcribed. So then we activated them with PMI-Namycin, which is a very potent activator, will purge out HIV RNA expression as well as other cellular gene expression. Then using these RNA-specific probes, which is 96 probes targeting 5' HIV of conserved regions, and another 96 probes targeting 3' HIV with another fluorophore. So when we fix the cells using FISH, which is fluorescent in situ hybridization, then we can see them on flow cytometry, then we can sort them.
Okay, so you're fixing them so the probes can get inside of the cells. Okay, and so what are you finding out from that?
So we found that in the past, so first there has been a previous report, for example, from Frank Matarelli and Lisa Franco, saying that in these clinical samples, HIV integrates into these very specific sites, but no, it's not integrating into these sites. It's just that if you take primary cells and infect them with HIV, you see integration all over the place in active transcript units. However, in these patient cells that they found that HIV integration are enriched in these very specific sites, for example, in BOT2 or MKL2, these specific sites, but we still don't know why. So where our question was, there must be an in vivo selection pressure that leads to the enrichment of these HIV-infected cells that with HIV integrated into these specific sites. So that's why we wanted to use our method to understand this HIV hosting action. So we pulled them out. We found that, for example, we found like HIV integrating into NFAT-C3, which is a gene related to T cell activation. HIV actually dominates over the original NFAT-C3 promoter, just dominates this and drives the transcription of NFAT-C3. And however, it produces a truncated or non-functional form of NFAT-C3 and sort of shut down the T cell activation. However, although it uses the splicing and skip through this HIV RNA genome to splicing to this NFAT-C3, the HIV genome expression remains intact. And then it's because HIV activates other cryptic splice sites to splice out all these RNA transcript required for active replication. So HIV is still being made, but T cell activation just shut it down. So we think that this may be a way that these cells do not die of maximum T cell activation. Because typically, if you have a replication content in HIV in the CD4 T cells, upon maximum activation, they will either die of viral cytopathic effects or immune clearance. But we think that because this HIV-driven aberrant transcription and alternative splicing that get these cells away from immune recognition or dying.
So this was work that you did previously and you brought on to Yale, right?
So this work was done in part at Johns Hopkins, but now at Yale I have two postdocs who are both working on this.
What other things are you looking at in your lab?
So this is one thing that we look at. We are actually looking at another other aspect, meaning that how can we use our molecular understanding to cure HIV? So it's not just to characterize things or for pure curiosity. From my standpoint, I was hoping if there is a way that we can use our knowledge to provide a treatment. So right now, the other direction that we're working on is the block and lock strategy, or is there any way we can stop this clonal expansion? So we have a screening system that we use 1,400 FDA-approved drugs to try to see if there are any drugs which can suppress HIV expression without hurting the cellular gene expression. And we found some hits. We're in the process of validating them in patient cells and look at their mechanisms.
Okay. So would you have any advice to someone who wants a faculty position and is in the audience now?
Oh, so this is a very interesting question. Long story. So first, it's a privilege to do hypothesis-driven research. It's this enthusiasm that keeps me working till now. For example, experiments won't work. 90% of them won't work. I have one whole year of things that doesn't work, and then I have one whole year of like empty gels. That's just common. However, you just keep thinking, why am I here? I'm here because I want to find a cure. Cure is not there yet. Okay, but I want to know how HIV does this and that it never stops surprising us. So it turned out that because this is the thing that I want most. I want to spend my life when I'm young and active working on the thing I'm most enthusiastic about, and HIV happens to be the one I'm really interested in. So this is a drive. And then, well, everyone has passion in this room, and then not everyone wants to do academia, and why is that? The reason we need a lot of help. If I want to go far, we need a lot of help with friends. If I want to go fast, I can go alone, but I get a lot of help. For example, I have a great mentor, Bob Slicano, who is just a wonderful mentor, guiding me all the way through. However, he started this training, like he trained PhD students, like postdocs. I've been I just joined his lab, like, Bob, I read all these literatures. I'm going to cure HIV. He's like, what do you want to work on? HIV latency. How are you going to work on this? I don't know. And then he's like, go back and think about this. And just keep back and forth for 2 months. I'm still sitting there reading papers. So it's like, I'm not going to read papers for 5 years. Then we came up with hypothesis-driven projects. And then so this is the start. And then the second thing is that the sky is the only limit, because I joined Bob's lab, I have no cell culture experience, no fax experience, never touched any virus before. You just learn it. If you can learn, ask your friends. And when I was a second-year student, there was a first-year rotation student, Bobby Buckite, I just learned P24 from him. It's okay. I was actually way older than my peers because I did my physician training. It's okay. I love to talk to them, talk to my younger colleagues and older colleagues. It's just great. So I learn from them. But by like fourth or fifth year, it's really the time to determine whether you will fit this research career, because you need to feel that I want to go to a lab every day. When I'm taking a bath, I'm thinking about my project tomorrow. That says that you should go to academia. And then I went on for this. So when I graduated, I was on this. So there's this NIH DP5 Early Independence Award. So they say, at the year you graduated, if I have this proposal that's totally brilliant, NIH will fund you, and you're an assistant professor. It's like, great. So I applied. I passed the Hopkins nomination, but when the grant gets to NIH, it says, you're just Bob's postdoc. You just sign off with Bob's name. It probably looks the same. So I was like, oh no. And then it's totally trashed. And I was like, Bob, I can't do research, and I'm just so sad. But I feel like I have some ideas I still want to do. He said, oh yeah, maybe. So then he very kindly promoted me to a faculty position. Then I wrote a R21, and then I got it. So writing a grant is key to these research, because it's not just the practice of writing. You need to plan a really good structure that you can persuade yourself, persuade the reviewers. But from that, I learned that I am going to not only just design my experiment tomorrow or this week, but I'm going to plan this out for 2-3 months. And I feel that grant writing experience is a really good start to see how we can get to this.
Well, we certainly do enough of it. I love your enthusiasm. It's great.
Thank you.
Did you get all that, young folks out there? Thank you. All right, we have about 10 minutes. John and Steve, I want to talk a little bit about the HIV reservoir, because you both work on that, and you've published together on it.
We work together.
Together. So tell us, first, what is the reservoir and what are the issues?
The reservoir is a pool of HIV. There's a touch of a controversy here, but I'll give you our view, which is the correct one. And that is, it was actually, in a sense, first identified and characterized by Bob Silicano's lab, by Yachi's thesis advisor and postdoc mentor. And it's a pool of infected cells, probably largely, if not essentially entirely, some kind of memory CD4 positive T cells, although it's possible there's some other types of cells, there's other subtypes involved as well. That's still being characterized. That contain HIV integrated provirus, and those cells, a few years ago, it was simply thought that this was kind of a stationary pool, that these cells, once infected, basically could live forever, and that they might, in fact, decay with time. And there was some data that suggested a very, very slow rate of decay, although it was so long that the virus would outlive the person that had it before it was down far enough to be gone. It's a very difficult problem to study, as Yachi pointed out. One cell in 1,000 has an integrated proviral DNA in it. And as Yachi's work showed, very nicely showed a few years ago, the very large majority of the proviruses that you detect by standard kinds of PCR assays or hybridization assays are in fact contain defective viruses that cannot give rise to reinfection. But in any individual who is on therapy for 10, 20 years or more, if you stop therapy, the virus comes back. And it comes back within a few weeks, typically. There's some variation in that, but a couple of weeks is the typical before you start to see it again.
So I remember you said this at Columbia when you visited. There's a random rate of reactivation.
Yeah, an important concept here is that the reactivation has nothing to do with stopping therapy. These cells are always, a small fraction of these cells are spontaneously reactivating, but it's clearly a very small fraction. But the numbers are although it's one cell in a million roughly that you can detect, and it's probably 10 to 100 cells in a million that actually have proviruses that are capable potentially of giving rise to infectious virus that are not defective in any way. And
so in a person on antiretroviral therapy, these reactivations occur continuously,
yes,
but they're suppressed.
You can detect them, and you can detect them in almost everybody, as my colleagues down our colleagues down in Frederick showed. You can detect them using a very sensitive PCR assay, you can, in patients, you can detect a little bit of low level of virus in blood. It's 10,000-fold or more below what it was before you started treatment, but there's a little bit there that's almost certainly the product of this ongoing constant reactivation. And as soon as you stop therapy, this virus can then start to infect cells again, having been completely blocked by the antiviral drugs you were giving, and back it comes. And it goes, remarkably, it actually goes right back to the level, essentially to the level where it was before you started therapy, even a few decades earlier. There's a memory in the system which is as yet not terribly well explained, but is really quite an interesting part of this.
So what, in 3 minutes, what are the issues that you deal with?
Well, how can you get rid of those cells, obviously,
Okay.
is the big issue. How can you, and before that, how could you identify them? Is there any marker, anything that you can use to identify the cells, or is there any other kind of strategy you could use? And a major strategy that's being tried is although it's the preliminary results are not wonderful with this, is to use some kind of activating agent that would cause the virus to get out of whatever latent state it's in. And the problem is we have really no idea what the latent state of the virus is in vivo. There are lots of in vitro models of viral latency where you infect cells and then the virus can go quiescent. But we have no idea which, if any of these apply into the in vitro, because it's such a needle in a haystack. But if the major idea is called shock and kill, that if you could activate those, then the virus replication itself would kill the cell, and you'd still have therapy present so that that virus wouldn't spread, or the immune system would kill the cell. And the few experiments that have been tried in patients with these have not yielded very promising results yet.
Any thoughts, Steve?
At least now, with the technology that's available to us, I'm somewhat pessimistic. The back of the envelope calculation suggests that there probably are millions of cells that are infected that carry live proviruses. And we also now know that, and Yachi mentioned this a little while ago, that in many cases, and perhaps in all cases, the cells that carry the live proviruses can clonally expand. So even if you knocked back their numbers, at least in theory, the numbers could be replenished because the cells are capable of dividing. And that actually posed a dilemma, an intellectual dilemma, in the sense that people had assumed that if you had a clone of cells of that sort, that all the proviruses and all the SIBs, all the related cells, would behave identically. And so either all the cells would not express virus, or alternatively, all the cells would express virus. And one of the things that our colleague Mary Kearney has shown is that that idea is wrong, and that within a clone, the cells can behave differently. And that actually also, I think, is a pessimistic piece of data in the sense that not only to sort of win the war, you have to kill all the bad guys. And if not all the bad guys, if there are probably thousands of clones representing millions of cells, and within a given clone, not all the cells and not all the proviruses, not all the viral DNA behaves the same way. It's a really daunting problem. And that's connected with the idea that in terms of what we often look at, because it's convenient, the amount of infected cells in the blood really represents about 2% of the total T cells and total infected T cells. And if we're going to somehow get at this very large reservoir and eliminate it, you have to have some way of either defeating the virus after it is released or getting at all of those cells. And I think given the current technology, that's a daunting task. Now, 20-odd years ago, 25 years ago, everybody who got HIV died. And we've now progressed to the point where with antiretroviral therapy, people don't lead entirely normal lives, but they certainly do enormously better than they did. So that was a very daunting challenge, and we've passed that. And it required the invention of new drugs, antivirals, which didn't exist. And now we're sort of to the next leg of the problem. And I hope my pessimism is ill-founded in the sense that I hope that these people, particularly the young people, can help us with what seems now, with what we know and the tools we have to be intractable, to become tractable, so that we can at least take patients off of the kind of antiretroviral therapy they're on for life now.
Well,
So far, if I could just add one quick
yeah,
thing.
of course.
So far from the kinds of studies, for example, Iachi is describing, the mantra seems to be that in this field, the better the science, the worse the news. The discovery that these infected cells can expand is really makes it clearly a much harder problem even than we'd appreciated.
So I'm one of the oldest people in this room, not the oldest probably, but certainly old enough to remember how this world felt before the fear of HIV came into being. And I lived through the early days of blaming it on this group or that group, and of course it turned out not to be true. But with the progress that you've made with regards to therapy, do you attribute the increase in AIDS now due to the attitude of people saying, well, it doesn't matter if I catch AIDS because I've got triple therapy that I can get, or is it some other reason that it's still increasing?
We should be a little careful with the nomenclature, I think. There's not an increase in AIDS. AIDS is...
I didn't mean AIDS, I meant the infection. I'm sorry.
So we have, but we have the following dilemma. We're doing a very good job in those who are treated of suppressing the disease and actually preventing transmission, because if you drop the level of virus in the person, they're not going to transmit. But that has, in some populations, engendered bad behavior. And that's an unfortunate bit of human nature. And as long as there are there's about 2 million new infections a year worldwide. And if we saved all those people, which we would like to do, I mean, I'm certainly, I've spent the last, 25 years of my life working hard to get this to work better. But you have to, in conjunction with keeping the people alive, we have to also take into account the problem of the spread. And there is a goal that you get to 90-90-90, where 90% of the people who are infected are diagnosed, 90% are on therapy, and 90% of those are in remission. And we're not anywhere near that in this country. And we have what is supposed to be an advanced biomedical system here. And we're nowhere near 90-90-90. In fact, some of the countries in Africa, in that sense, are doing better than we are. So we have a research problem and we have a public health problem, and they're both serious.
And a not insignificant number of people in the U.S. still die of HIV infection.
All right, we are out of time. It's unusual for TWiV. We usually are not constrained, but we have to wrap this up. You can find TWiV at microbe.tv/TWiV. You can send questions and comments to TWiV@microbe.tv. If you like what we do, support us financially. If you give $1 a month, it would really help with our production expenses. You can go to microbe.tv/contribute to figure out how to do that. I want to thank my guests today from Tufts University, John Coffin. Thank you very much.
Thank you.
From the NCI, Steve Hughes.
Thank you very much.
From Yale University, Yachi Ho.
Thank you.
Thank you and good luck.
Thank you.
From Rockefeller University, Matt Takata. Good luck and thank you.
Thanks.
And of course, from parasiteswithoutborders.com and the Living River, is it a .org? Or
Yeah, it's .org.
That's his fishing website. Thank you, Dickson.
Trout stream ecology, please, it's science, not fishing.
Dixon Despommier, thank you very much.
It's a pleasure to be here.
I want to thank the organizers of this meeting, particularly Theodora Hatsuano, for suggesting we do a TWiV here. Should we come back next year? Yeah. I'm Vincent Racaniello. You can find me at virology.ws. I want to thank ASM for their support of TWiV and Ronald Jenkins for his music. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.
Thank you.